Hi, my name is Gjoni. I'm a third-year PhD candidate in the Pharmaceutical Sciences and Pharmacogenomics program at UCSF in the lab of Catherine Pollard. My research focuses on genomic variants that disrupt 3D genome folding, leading to changes in gene expression that contribute to disease. The spatial organization of DNA into its 3D structure facilitates gene regulation by guiding the contact of gene promoters and their distal regulatory elements. This is often done through topologically associated domains, or TADs, which are structures within which contact is much more frequent than outside of. The reorganization of TADs can change the frequency of genomic contact between distal regulatory elements and cause changed transcription. This mechanism has been shown to lead to various developmental disorders and cancers. For example, the formation of a new TAD boundary within the SHH TAD lowers the rate at which the gene promoter contacts its enhancer, resulting in its downregulation, which leads to limb malformations. The opposite of this mechanism occurs in lymphoma, where the loss of a TAD boundary allows the MYC oncogene to gain contact with the new enhancer and become overexpressed. Autism spectrum disorder, or ASD, is a great candidate for this mechanism since up to 80% of cases don't have a genetic diagnosis, given that most variants have an unknown effect. We hypothesize that disruption of genome folding is a prevalent but overlooked mechanism in variants in ASD. Experimentally testing this requires genome editing and chromatin capture experiments, which are cost prohibitive to perform at scale across a whole cohort. To overcome this barrier, we use a machine learning tool developed in the lab called Akita that can predict chromatin interactions in the form of high-resolution contact frequency maps from genomic sequence alone. Using Akita, we can compare predicted contact frequency maps from sequence with and without each variant found in individuals with ASD and generate disruption scores that quantify the changes in genomic contacts in a 1 megabase region surrounding the variant. By incorporating epigenomic data from excitatory neurons, the cell types most commonly affected in ASD, we can generate weighted disruption scores that should be more representative of the affected tissue. Using this pipeline, I've prioritized candidate variants that are predicted to disrupt regulatory interactions of genes implicated in autism. To test our predictions, we plan to use an in vitro model of excitatory neurons generated from genetically engineered induced pluripotent stem cells. I hope that my research will help explain a new aspect of these disorders and will improve our knowledge of their disease mechanisms and eventually of how to build therapies. And more broadly, my goals in graduate school are to gain expertise in the relationship between genomic variation and disease phenotype using bioinformatics. The ARCS Award will help me achieve these goals by providing me with more freedom and access to tools that help strengthen my research. And I'm very excited and honored to be a part of the ARCS community. Thank you.